All right, guys, many of you have been asking for a study guide for exam one, record a video that you can get on and just look and make sure you have all the questions answered in case you miss study group. So I'm going to go as fast as I can and still hit most of the questions. Label a plant cell. You have to know most of the main parts: mitochondria, chloroplast, Golgi apparatus, the endoplasmic reticulum, nucleus, central vacuole. That's 6. You have to know 5 of them. So make sure you look it over and just be able to identify the main parts. Next question, list 2 differences between a eukaryotic and a prokaryotic cell. Well, eukaryotic cells have a membrane-bound nucleus. Prokaryotic cells do not. Membrane-bound organelles are also found in eukaryotic cells, which allows them to be much bigger. Prokaryotic cells lack membrane-bound organelles and are a lot smaller. The cell size is limited if it's a single cell. This is because cells cells very quickly have to move materials into and out of them. If they become too large, if the surface area is not large enough to maintain quick diffusion of materials into and out of the cell, they will become backed up and won't be able to perform the chemical reactions that they need quickly enough for them to survive. So their surface area to volume ratio has to be really large, with a large surface area and a small volume, so that they can quickly diffuse needed materials like sugars in and get rid of waste products and move them out. Number 4 is define hypothesis. A hypothesis is just an educated guess. Number 5, 3 of the 7 characteristics of all living organisms. So organisms have to grow, they have to respond to their environment, they change over time, they perform chemical reactions, maintain homeostasis, reproduce, and they have heredity information that they pass on from one generation to the next, have a genetic code. Pick 3 of those or 4 of them that you think you can remember for the test. Rough ER, Golgi apparatus, and plasma membrane are involved in digestion through the lysosomal pathway. Basically, as food gets brought into your body, it will pass through the bloodstream and get absorbed into cells. And the way this happens is through phagocytosis. So the plasma membrane will kind of wrap around your food and make a vesicle and pull that food inside through phagocytosis. That vesicle will then join with the lysosome. And in that lysosome are protein enzymes that will be responsible for breaking down the food. Now, how do the protein enzymes get there? Well, the ribosomes made them. The rough endoplasmic reticulum helped to modify them. And the Golgi apparatus further modified and packaged and sent them to the lysosomes, where they were ready and waiting to aid in your food digestion. When that food gets broken down, it gets recycled back into the cell. Those pieces do, and may be made into new proteins that will go through that whole process again. List the 4 major biological molecules. Well, we have carbohydrates, lipids, proteins, and nucleic acids. And you have to pick one of those and list 2 characteristics of each. So pretty much all of them, except for nucleic acids, are involved in structure of the cell in one way or another. Lipids are involved in the cell membrane. Carbohydrates also function some in the cell membrane, and proteins help in the cell membrane as well for the function or for the structural parts. Nucleic acids are your genetic information, and they also are energy. ATP is a nucleic acid that's energy. Carbohydrates are short-term energy, lipids are long-term energy, and proteins act as enzymes, and they help to catalyze metabolic reactions. You will be given a scenario and you will use it to do the following. So an example I might have on the test, or something very similar, is you want to design an experiment in which you want to know how the amount of light affects plant growth. And then from that scenario, you have to answer the following questions. You have to tell me one hypothesis you would test. So maybe I might say, I want to test the bulb wattage. So I think that bulb wattage, my null hypothesis would be, I think that bulb wattage will have no effect whatsoever on plant growth. They're all going to grow relatively the same. And then my hypothesis would be, I predict that the 75 watt bulb will have much higher plant growth than my 40 watt bulb. So you could come up with that. my control for my experiment might be, it's kind of a hard one. You might pick something that's kind of in the middle, maybe a 60-watt bulb, or you might say just regular room lighting in like a fluorescent light, no added extra lights if it's like in the school, or maybe UV light outside, whatever the natural light is compared to artificial light. So you might be testing that type of light. 2 factors that must be kept the same in the experiment. So you might say that the amount of water has to be kept the same, the amount of soil, the type of plant, the temperature of the room, all those things. You would have to pick 2 factors as you're designing your experiment. Independent variables, what you're changing. So in my scenario, my independent variable, I am changing the amount or the wattage of my light. Label the columns in the data table. So your independent variable is going to go in the left-hand column. So that would be the amount of light in wattage. Make sure you write your unit. And then your dependent variable, or what you're measuring change in, would be your plant growth. So that would probably be in centimeters. Make sure you write your unit down. The next one, it says you'll be given a scientific experiment write-up and graph. From that information, surmise the independent variables. Again, independent variables, you are looking for what that situation is testing for change in. What are they changing in it? Oh, I said that kind of wrong the first time, but what are they changing? The independent variables is what they are controlling. And then the dependent variable is what they're measuring that change in. Okay, so you got to look for those things as you read a paragraph and then look at the graph. It says which axis of the graph represents the independent variable. The independent variable is always going to be on your x-axis. It's always going to be on that lowest axis, the bottom axis. And your dependent variable is always going to be on your y-axis. So your categories, if it's a bar graph, go on the x, and then your amount of change, or your amount that you're measuring, is on your y-axis. And you must also answer questions using the graph. So that's kind of like, can you follow instructions and look on the graph and tell me which one's the best or which one's the worst, those kind of scenarios. You will be given a paragraph about a scientific experiment. From that information, you must pick out, again, the independent variable, what are they changing, the dependent variable, what are they measuring, 2 constants, what are they keeping the same, and their hypothesis. So what did they think would happen? So there's a rundown on your short answers on your study guide. Oh, there were the 2 that I mentioned that you had to look at and do. Let me pull up the actual test because it's on there. It was write out the chemical reaction for cellular respiration that takes place in the mitochondria and the chemical reaction for photosynthesis that takes place in the chloroplast. If you just write the words for this one, that's okay. That was at the very end of my notes. So from the lecture part. So that is, in the mitochondria, you have glucose, or sugar, plus oxygen, yields energy, carbon dioxide, and water. And in the chloroplast, to make that sugar, what we're doing is we're doing the reaction backwards. So you're taking carbon dioxide and water in the presence of light energy, and that yields sugar, or glucose, and oxygen. And then how is photosynthesis and cellular respiration equation compared? One is basically the opposite of the other. So there's your study guide rundown. I hope that's helpful. I'll do another video here in just a little bit about the multiple choice questions. Have fun studying.